sell me on this idea of metabolomics, and I agreed with him on one condition. I would not work with any invasive samples. No blood, no tissue. I wanted to take this in a different direction. And so I threw a few different ideas at him. I told him, well, why don't you look at tears or hair or spit or something? And somehow he misheard the word spit as sweat and packed me off to a dermatologist to discuss the project in that sense. So over the last couple of years, I've been working with Dr. Raja Sivamani over at the Med Center in Sacramento. And when I first met Raja, the question I put to him was, has anyone looked at sweat in the context of skin diseases? And surprisingly, the answer was no. Now, sweat shows up on the skin surface. It has good equilibrium with the cell surrounding the sweat gland. So you'd expect a lot of good information. A lot of these compounds, you'd expect to potentially see those in sweat. In addition, sweat exists in equilibrium with blood, so it'll give you some really good systemic information that we could push in a non-invasive manner. So it seemed like we hit the jackpot. The only question was, how do we actually prove our point? And we decided to look at a couple of different skin diseases and ultimately ended up choosing atopic dermatitis, or it's commonly known as eczema, but let's move on from that one for a bit. Now, atopic dermatitis is an inflammatory skin condition that affects mostly kids within the first 6 months to a year of life. In some cases, it resolves by the time the patient reaches adulthood, but in a lot of cases, it will persist well into adulthood. It's characterized by a number of different things. Subjects with atopic dermatitis have dry, scaly, pruritic, or itchy skin, and they go through these periods of ebbs and flows of inflammation, known as flares. There's not a lot known about the disease in general. There are some hypotheses as to its pathophysiology and its etiology. And unfortunately, because there's not enough known about it, how it starts and how it progresses, treatment options at this point are relatively simple and mostly symptomatic treatments. Either creams to improve the skin barrier and prevent dry skin, or steroids to prevent inflammation. And given these two major hallmarks of the disease, the sort of the dry skin barrier and the sustained periods of inflammation, there were basically two major hypotheses that originated as to its pathophysiology. The first is this idea, sometimes referred to as the outside-in hypothesis, where you have either genetic events, environmental events, any number of different factors that basically alter skin barrier. This, of course, leads to ingress of haptens or allergens, pathogens, what have you, and somewhere down the line, you end up with inflammation. Now, on the flip side to that is the inside-out hypothesis, would suggest that you start with some sort of inflammatory event. This is mostly mediated by Th2 helper cells, or IL-4, the cytokine. That decreases lipid production. And that, in turn, alters the skin barrier. And for a long time, these two hypotheses were in competition with each other, up until about, actually, 8 or 9 years ago, when Peter Elias down at UCSF said, well, hang on, why don't we just combine them all and essentially come up with this whole idea that the process is cyclical. You have an environmental or genetic insult that alters the skin barrier, leads to inflammation, which in turn will come back and continue to perturb the skin barrier, essentially leading to a vicious cycle that cannot be broken. And to my mind, I think that makes the most sense, because subjects with the disease do tend to go through these ebbs and flows of inflammation, so the whole thing is probably just one giant cycle. And unfortunately for us, most of the research done on this pathophysiology, or any studies done on atopic dermatitis, focus on very small parts of this. They either focus on the genetic basis, the environmental basis, what's going on with IL-4, what sort of lipids are involved, but they're all very, very targeted, and so no one's actually taken a step back and tried to understand this pathway as a whole, which is where we decided to try and attack it from. John's already talked about this idea of lipid mediators that can affect inflammation, for example. Now, one of the compounds he talked about, the ceramides, are actually a very, very key component of the skin barrier. If you look at the uppermost layer of skin, or the epidermis, the stratum corneum, which is the uppermost layer of the epidermis, is actually consisting of little bricks known as keratinocytes, which in the middle is filled with essentially a lipid matrix. Now, about 50% of this is ceramides. So not only do they play a role in signaling and inflammation, they're actually a key component of the skin barrier. So understanding how those are affected in an atopic dermatitis system, I think, would be key towards elucidating the pathophysiology. John's already also mentioned that all of these mediators interact with each other. To give you a really, really simplified schematic, let's take arachidonic acid down here. So SN2 arachidonic acid, which is its precursor, is metabolized by phospholipase A2 to form arachidonic acid, which can be metabolized, say, by cyclooxygenases to a variety of products, say, for example, prostaglandin E2. Now, prostaglandin E2 is a known inhibitor of lipolysis, which ultimately drives ceramide production. Ceramides not only affect the skin barrier, but through a variety of mechanisms can actually induce phospholipase 2 activity, again, re-regulating this cycle. Then up here at the top, we've got the cannabinoid receptors, C2, which can inhibit adenyl cyclase, which actually regulates COX activity. So looking at each of these in isolation, you don't get the full picture. But if we can profile these collectively, as we have methods to do so in the lab using LC and SMS, we can really start to appreciate various aspects of this entire pathway, and hopefully gain a lot more information about the pathophysiology of the disease. And maybe we can identify some new therapeutic targets that could be used to either treat the disease, if not maybe cure it down the line. But all of that has to do with skin. Where does sweat come into this? Most of the samples that are used for atopic dermatitis are skin biopsies. Now, if you want to, what a skin biopsy is, it's basically pulling a plug of skin from some surface on your body. Typically, these are about 4 to 8 millimeters in diameter. And I don't know about you, but if I had to come in and get skin pulled for me, every 6 months to a year, I wouldn't be a very happy camper. I suppose the other thing you could do is go look at blood. Not a bad idea, but I'm scared of needles. I don't like blood. I don't like getting blood tests done. So why not look for something a little more non-invasive? As I told you earlier, we look at the architecture of the skin itself. You have the epidermis up here. This sort of section down here is the dermis. It's packed with fibroblasts. And through these fibroblasts, you have the sweat gland. So where some of these lipid mediators that we tend to study can be produced in the fibroblasts, they could diffuse over into the sweat, either through passive or active mechanisms. In addition, the sweat gland is relatively closely associated with the skin capillary network. So you could have diffusion of circulating mediators back out into the sweat gland. So in this respect, you get information not only locally at the site of inflammation or atopic dermatitis for our purposes, but also globally. So sweat sounded like a really good idea. That, of course, precludes the question of how do we collect it. I suppose there are a few different ways. Get a guy up here giving a talk, he's going to be sweating bullets. That's one way to do it. I suppose you could also just go down to the arc and towel people off as they come off the treadmill. or you could stick them in a sauna, which has actually been done, believe it or not. But those are relatively uncontrolled mechanisms of collecting the sweat, and there's a risk that something like exercise or heat shock would essentially induce a lipid mediator response, which would affect the quality of our results. What a lot of people do in forensics is actually put a patch on someone's arm, an absorbent bandage of some variety, let them loose for a week, and then bring him back in and collect the bandage and examine the contents of that. That's actually not a bad idea, except for the fact that your skin produces one other major secretion, the sebum, the oily stuff that leads to acne, among other things. Now, telling what's sweat and what's sebum on that patch is relatively difficult. So for our purposes, where we're trying to focus exclusively on the sweat, it's not the best thing to do. So actually, about 10, maybe 15 years ago now, a company out in Utah called Westcor developed this little device called the macroduct sweat collector. What it is you've got a battery pack here connected to 2 electrodes containing the drug pilocarpine. Pilocarpine is typically used to treat glaucoma because it produces intraocular pressure, or it counteracts atropine. So when you go to get your eyes dilated, for example, and they use atropine, and if you put a couple drops of pilocarpine after that, your eyes would actually contract back to normal. So so pilocarpine is an M3 muscarinic agonist that, when infused into the skin, actually stimulates sweating locally at that one site and nowhere else. So what we could do is use electrophoresis to force pilocarpine past the skin barrier to trigger sweating, for example, at the forearm, maybe in this region here. Then what they've got is a little collector. So this is sort of back view of it. It's about the size of a wristwatch, and there's a little tiny hole over there. And you can strap it onto the arm. In the front, you've essentially got this little sort of coiled tubing. So the sweat wicks up through that little hole, collects in the coiled tubing, and at the end of your collection period, you can simply cut out the piece of tubing and squeeze the sweat into some sort of collection vial. So that way, you've got a measured volume of sweat that you can analyze, and it's in a convenient form to run through an LCMS, for example, to obtain information about our various mediator profiles. Now, Westcor developed this for cystic fibrosis diagnosis, since that is the most common application of sweat in a diagnostic sense. But about 2 years ago now, a group out of the University of Cordoba in Spain put out a review article essentially asking, why has no one done metabolomics of sweat? They've since followed it up with 2 studies in which they first demonstrated the presence of various small molecules, amino acids, some fatty acid derivatives in sweat. And actually about 2 months ago put out a paper showing the potential for sweat diagnostics in screening for lung cancer using an amino acid-based approach. So look, all of these sort of serendipitously happened right around the time this project was under development. And that's how we came up with the idea to run metabolomics of sweat to try and understand more of what's going on with the idea of atopic dermatitis. So our overall goal is essentially, of this project, is to establish sweat as a diagnostic matrix for inflammatory skin disease. And in so doing, understand more about the pathophysiology of atopic dermatitis in order to ultimately develop more therapeutic targets and also just understand what's going on with the disease. We're operating under the hypothesis that our various lipid mediators that we measure, predominantly the oxylipins, the endocannabinoids, and the ceramides, are involved in some way in the progression of atopic dermatitis. These interactions are exaggerated during the inflammatory flares. They're mediated by the therapies used, predominantly corticosteroids, and all of that can be non-invasively assessed using sweat. How are we going to go about doing this? The first step for us is to actually validate the use of that collector for our purposes. When it was developed for cystic fibrosis diagnosis, I'm sure Westcor actually went ahead and validated it for that use, but we don't know what's in that collector. Is there stuff that could contaminate our sample, rendering it completely useless? Will our analytes end up sticking to the tubing, and therefore we won't actually see anything? These collectors typically come with a spot of dye just to the side of the collection orifice, which is used to make it easier to visualize the sweat. Well, that doesn't affect chloride measurements with CF. The dye might contain stuff that, again, interferes with our assays. So the first step is to actually understand all of that. And then the bigger question, is there actually anything in sweat, or are we just wasting our time with the rest of the study? Fortunately, about 40 years ago now, excuse me, a guy named Lars Forsgren out of the Mayo Clinic did a study of prostaglandin E2-like compounds in sweat, and actually found some fairly significant concentrations. So that suggests we're not completely barking up the wrong tree, but that's only one analyte. What else is in there? So we do have some really cool pilot data. For example, we know now that dye actually is very, very rich in lipoxygenase metabolites, one of the 3 arms that John had up earlier. So we probably don't want to be using that. The collection device itself is actually fairly clean. Most of any sort of contamination that may or may not be present is well below our calibrators, so that suggests that the device itself is fairly viable. And we haven't quite yet assessed whether or not we're losing any analytes, but that's in the works. The other thing to consider is when's the best time to collect the sweat. Since sweat does take in input from the blood as well, in addition to the skin, there's this idea that diet might end up affecting the levels of some of these compounds we see through metabolism of available substrates like linoleic acid or EPA. So to do that, what we're going to ultimately end up doing is sampling people before and after they've eaten a standard meal, and essentially seeing are the levels of these compounds different, and if so, which might be the better time to sample them. That in itself will also give us information about what's actually present in sweat for the purposes of our testing. We sampled a few people after eating, actually, and what we found is a fairly broad variety. Out of the sort of 125 odd different mediators that we study in our lab, we found about, I think, 40 of them. So there's a bunch of stuff, and they represent different aspects of arachidonic acid metabolism, endocannabinoid metabolism, and cervical metabolism. So it's promising. And the final thing is the temporal stability of these compounds, because you find it once, great, but what if 1 week down the line the levels are completely different? And so to do that, we're actually doing some stability studies where we're bringing the same subject 3 different times, at least a week apart, and seeing how similar or how different the levels of our compounds of interest are. So that's the first thing, actually validating the use of this collection device and proving to ourselves that sweat will actually contain our compounds of interest and can be used as a diagnostic matrix. The second aim to that is understanding what impact atopic dermatitis has on sweat. Through studies in skin biopsies, mostly done in the early 90s actually, or actually late 80s, early 90s, we know a few different things. We know that, let's see.
You could raise your name.
Yeah, I might have to go down that route. Well, people know who I am. We know from skin biopsies that we have increases in prostaglandin E2, 12 heat, which is LOX alcohol, and LTB4, which is a 5 LOX leukotriene. All of these are in subjects with atopic dermatitis. We also know from plasma about 5 or 10 years ago now that there's an increase in LTB4, again, in subjects with AD. So we know that these mediators are involved, and the question is what's going on in the sweat. The other problem is those were very, very, very targeted analyses. They only had standards for the 3 compounds up there. What other compounds are involved in our profiles, and how are those interacting with in atopic dermatitis? We know that a large number of those compounds are pro-inflammatory in other disease states, cardiovascular disease, in metabolic syndrome, etc. what do those do in atopic dermatitis? So to do that, we're actually going to assess our various profiles in not only the sweat, but also the skin and in plasma. That way we understand what's going on at the skin surface level, what's going on systemically, because atopic dermatitis is actually symptomatic of a larger systemic dysregulation, and I'll go into that in a couple of minutes. And also, where does sweat fit into this paradigm? Are the markers we're seeing in sweat reflective of what's going on simply over here in sort of the skin? Are they reflective of what's going on over here? Is it a mixed signal? Where does sweat fit into this paradigm, and how do we actually appreciate the results that we're seeing? So that's the second aim. And the final aim is to understand what goes on when inflammation strikes and how is that related to the levels of our compounds. So we bring in subjects when they're in an unflared state, sample them once for their sweat. Let them go until they call us up and tell us they're flared. Bring them in again and sample them. And then actually treat the flare. Give them their standard corticosteroid therapy and watch what happens to those mediators. So essentially what we're asking is, which mediators are affected by the flare? Is steroid treatment actually doing anything for it? And does it bring it back down to normal levels? Now, the reason we're asking this question is we want to evaluate the efficacy of corticosteroids in general. Corticosteroids, and John, I think you'll agree, are a sledgehammer approach. They essentially wipe out arachidonic acid production and therefore anything that comes after it. Now, there are a lot of anti-inflammatory mediators as well that are metabolized from arachidonic acid, so potentially by wiping out arachidonic acid in general, you're wiping out the anti-inflammatory mediators with it. So if it turns out that it is only, those 3 and maybe a couple other mediators that are actually affecting atopic dermatitis, why would you wipe out the other 500 and how many ever others there are? Especially, we know that there are specific inhibitors for each of these that could be used instead. So that's what we want to understand. There are some other therapeutics out there for atopic dermatitis. For example, Xylutin is a 5-LOX inhibitor that would prevent LGB4 formation. PGE2 could probably be prevented by any sort of anti-inflammatory, ibuprofen, for example. And 12-HEAT. There are no good 12-HEAT inhibitors that I know of, but actually one that came out from a discussion with Dr. Sivamani was curcumin or turmeric. So this is purely anecdotal, but for example, women in India tend to put a lot of curcumin or turmeric on their skin to essentially for beauty treatments, I suppose. But comparatively, the rate of atopic dermatitis in India is about half of what it is in the US. Now, that is a huge stretch, but there might be something to that. So finally, I made the point that atopic dermatitis is probably symptomatic of a larger systemic dysregulation. Atopic dermatitis is really the first step in what's referred to as the atopic march. People with atopic dermatitis down the road end up with a variety of diseases, predominantly food allergies, asthma, and hay fever. So the question is atopic dermatitis really just a disease of the skin, or is there a larger systemic dysregulation going on? And can we use that information to maybe treat all of them in one shot? And that's really why in aim 2 we're looking at 3 different matrices to appreciate where things are going. So, what we're hoping at the end of all of this is to achieve 3 different things. The first, we want to establish sweat as a non-invasive diagnostic, and hopefully start moving the field of medical diagnostics towards these more non-invasive samples. The second, we want to understand the pathophysiology of atopic dermatitis in order to hopefully develop better and more targeted therapeutics for the disease. And the third, and this is really not a benefit to us as much as it is to the company, but the idea that Westcor, which has predominantly been producing its devices for the diagnosis of cystic fibrosis, suddenly opens up this whole new market in the field of metabolomics that it could then exploit, because it looks more and more that cystic fibrosis diagnosis is moving in the direction of genetic testing. So, with that, that is about all I have to propose before you, and hopefully by the time the symposium comes around in March, we'll have some really good data to show you guys. But yes, thanks very much for your attention.
Ellen?
Yes, it seems like one of the limitations of sweat side, of course, is that all you can see is the water soluble stuff. But a lot of the most important compounds you're talking about are lipids and derivatives. So I was curious, do you have any plans to look at the sebum as well?
So, Dr. Subramani's interest is actually in sebum metabolomics, and we are collecting sebum from these subjects as well. There are plans to eventually incorporate both profiles into it. That said, they have found some lipids in sweat before, not particularly water-soluble ones either. So there's this whole idea that sweat may not just be salt water as classically thought of, but actually a much more complex matrix.
Yeah, so I was wondering about the role of the skin microbiome and how, bacteria on the surface of where your collector is might be changing or somehow altering the compounds that you're picking up. And if people have thought about that.
Actually, that's one of the latest topics that have come up in this whole idea of skin biology. Yeah, there's definitely a microbial component to it. For example, a lot of folks with atopic dermatitis have increased Staph aureus colonization on lesional sites. We're precluding most of that by actually wiping the skin with alcohol prior to collection, so we're hopefully killing off all the microbes and sterilizing our site.
It kind of would be interesting to do a metabolomics/metagenomics study, like see what you get metabolite-wise, and also at the same time, instead of wiping stuff off, just let like your little collector scrape up some bacterial glue or whatever's there.
It's an interesting idea, but it's a very complex paradigm that you're talking about, because we know that physiologically the structure of the matrix of the skin is changed. So you would almost suspect that you might see different colonization, and it's still a cause-effect issue. So you could very well see different microbial colonization that may have nothing to do with these mediator effects. But it is an interesting idea.
We could collect from a non-eczema site, from an eczema site, excuse me.
True, yeah. And it'd be interesting if anybody's done this.
I think it's early stages for all of this, so.
I was just wondering, this genetic thing,
Yeah.
in our family, my older sister had atopic dermatitis, and then both of her sons ended up with atopic dermatitis, but her daughters didn't have it, and none of there's 5 girls in the family, and nobody else had it. So that's I think that there is some genetic factor here that.
Oh, absolutely. A lot of people tend to implicate the protein filaggrin. So filaggrin is a key component of the membrane of keratinocytes, and in about 15 to 20% of cases, there's a defect in the gene that would synthesize filaggrin. And so that's what a lot of people are attributing to the genetic basis, but I'm sure there are others. And in fact, one of the key diagnostic criteria to call it atopic dermatitis versus just some sort of inflammatory skin disease is the family history of the disease. So it's definitely something there.
I have a question. You haven't really touched on just the components, currently known components of sweat, just its general properties at all. And I would imagine that would have to at least warrant some sort of consideration as you're putting it into your LC-MS, right? Can you talk about the basic components, ionic strength, stuff like that? Do you know anything about that?
Not a whole lot. I mean, from what is classically known, sweat is just 98% water with some salt in there. But more recently, like I said, they found stuff with proteins and some fatty acids, other small molecules, and metal ions. We haven't noticed a problem yet in our LC. Our extraction procedure is actually relatively simple. We simply evaporate this. We add our deuterated surrogates to the sweat, evaporate the whole thing off, and reconstitute it in our internal standards. And we haven't noticed any major suppression. So whatever components there are, they're probably relatively minor in the overall scheme of things.
Oh yeah, I was just curious about like location where you would select it, and if you're planning to like look for an optimal part of the skin, since there's so much different biology going on.
Yeah. So atopic dermatitis lesions in adults, at least, which is sort of the population we're targeting for now, before we move this into a juvenile population, typically occurs on the inside of the elbow, the wrist, and corresponding locations on the leg, back of the knee and the ankle. So where we're doing our collections is actually sort of the classical site for skin measurements, which is the volar forum, basically about the first half of this area. There are a number of reasons why we're picking that site. One, it is sort of a classic site, so it's easy to go between studies. Two, the area is relatively sun protected relative to the upper surface of the skin. And three, there are actually minimal sebaceous glands in this area, so we decrease potential for sebaceous contamination. What else?
Well, then you're all done.